Well, hello everybody. This is Mr. Billick speaking to you in the past. I am not sitting in the classroom. Surprise. You should have already known this unless you were taking a nap or just cooking food during our meetings. Both is a distinct possibility. So, this video is going to explain what you're going to be doing today. I'm going to in my mind, I'm going to break it down into a certain ways. But first thing first, just like always, we have our starter question. All right. So our starter question says, use the allele key to the right to answer the following questions. Give the genotype for each of the following. Number 1, homozygous dominant. So I want the genotype for homozygous dominant. Number 2, I want the genotype for heterozygous. And number 3, I want the genotype for homozygous recessive. And I would like those 3 genotypes. You can pause it, but I'm just going to keep talking if you need a moment to think about that. The agenda today is I am going to talk about dihybrid crosses. I'm going to give you some practice. And then you're going to have to do some good old classic busy work for the sub. So you are expected to, it's going to be a gizmo and a doc, which I will explain in one video as well. The announcements is we are going to have a test. Okay, so you have less than a week. Next week, there is no Silver Day. So you will be taking the test on Wednesday. Wednesday's the 17th. The study guide is posted. Don't know what else to say on that. I'll tell you, you don't have to do the study guide. I want you to be prepared, though. All right. I will be back on Monday. Also, too, on Monday, for anybody who loves the seat that they're in, remember, Monday's the first day back where we could have a full class. And remember, Monday's the first day also. The first and last day, you get to pick your seats for the rest of the year. So you better be ready to go in and get right to your seat come Monday. Because where you are is where you're going to be until May, whatever, 14th. 20th, whatever day we end on, okay? So just kind of keep that in mind as well. All right, so we're just going to keep on trucking, all right? So what we're going to be, what I'm expecting you to do is just, for the most part, I want you to stay with me. Watch me explain these slides. I will then explain what you're going to be doing after this. But we are going to start talking about the dihybrid crosses. Dihybrid crosses are different from the monohybrid, monohybrid cross. Mono meaning one, so we are only dealing with one trait. Di means 2, so we are now dealing with 2 traits. Okay, dihybrid cross is when 2 traits are together. The traits are not linked, meaning they are not on the same chromosome. For example, we could have a phenotype that is hair color and height. All right. And what does that mean? All right, so a couple examples of a dihybrid cross, and you're going to have your allele key over here. We have our uppercase A, which means brown hair, our lowercase a, which means blonde. Remember, uppercase means dominant. If you don't know that now, I don't know how else to tell you. Uppercase is dominant, uppercase B means tall, lowercase b will mean short. So, how do you read a dihybrid? It's just like we read before, but just imagine that like you're only reading one section at a time. Don't get confused with all these together. So we're going to read this section right here. We have a capital A and a lowercase a. That's it. Okay? So a capital A and lowercase a, you already know if we have a dominant trait. So this means we're going to have brown hair. Then we just move to the next the genotype. Capital B and lowercase b. Capital B means we're going to have tall. So remember, if we have a dominant trait, a dominant allele present, that trait will be expressed. So when you look at this and you look at the allele key, you should say, this person or thing is going to have brown hair and will be tall. Now we just move on to another example. 2 lowercase a's. Those are 2 recessive alleles. That means we're going to have blonde hair. And 2 lowercase b's. So lowercase b's means you're going to have short. So blonde hair and short. The end. Okay? The last one, we'll just do one other example. Genotype would be capital A and capital A. If we have 2 capital A's, that means we are going to have brown hair because we just need 1 capital A for it to be expressed. 2 lowercase b's means it will be short. So, brown hair and short. Okay, just remember, just break this into one part at a time. All right, so those would be an example of hair. That would be an example of a dihybrid example. All right, so what we're also going to do is you're also going to have to pay attention on this. And I know that's difficult for you all sometimes, not all, majority, and we'll just say that. So, what I want to do is I'm going to kind of just go over just one little quick example that I already have pulled up. So we're going to be breaking down a dihybrid into a gamete, which is sex cells. Okay, so you're going to need to pay attention to here. So my example says here we are. A female rabbit has the genotype GG. Well, you want to know what? I don't like this one. We're going to go to this one. A male rabbit has a genotype of, and I'll zoom in, capital G, lowercase g, capital B, lowercase b. So I'm going to write this out here. G, capital, lowercase, capital, lowercase. All right. We are now going to take this and divide this into sex cells, okay, into gametes, for this case, since it's a male rabbit, we are going to divide this into 4 sperm cells, okay. Remember, I want you all to kind of think back to the idea of meiosis, and this is why it's connected. Remember, in meiosis, we have this part is from your dad, so you have half of your DNA comes from your mom, and half of your DNA comes from your dad. It's the same idea in a rabbit, okay? The rabbit's mom is half of its DNA. The rabbit's dad is half of his. Remember, so we started with something like that in the beginning of meiosis, and then our final product was something that was half of that. So we had some information from your dad and some from your mom. And that was the final product. And remember how I said, okay, then that would make 1 of 4 sperm cells. So this is me explaining the process kind of behind this, all right? So keep that in mind. Just like rabbits, like I said, or humans, rabbits have half of their information comes from their mom and half comes from their dad. All right? So I want you just to imagine, we're just going to split this in half here. This information is coming from his dad, and these traits are coming from his mom. Okay? So, 50% makes 100% of this rabbit. All right? We remember, we are now trying to, the end product of this is we're going to have 4 different sperm cells, okay, that are going to have the same thing. They're going to have half the information, 50% is going to come from the dad, and 50% is going to come from the mom. Okay? Fart. Okay? So we're just going to say this half, I'll just make it simpler. This half is dad. This half is mom. This half is dad. This half is mom. This will make sense in a moment. Dad and mom. Okay. So, each one of these sperm cells now has to have at minimum, it's going to have It's going to have one letter from mom and one letter from dad. And I'm going to show you how we're going to break this down. So, first things first. We're just going to start right here. So we're going to have a capital G. That is the trait coming from dad. So we can put one G here. Okay? So we have a capital G here. Now we need to figure out, okay, what is mom going to bring to the table? All right? So mom, we can say we're going to have G. Capital G could go with uppercase B. Boom. We have one gamete. I want us to remember this too. And maybe I'll kind of, let me go back. And I'll kind of do this in colors. So maybe this might make a little bit more sense. This is coming from your dad. I know it's pink and blue. It's classic boy and girl colors. So that G information came from your dad. The B came from your mom. Half and half. Okay. Now let's kind of go back a little bit now. Now we can also have G right here. We're still figuring out all the different combinations. Can also be paired with the lowercase b. All right. So the information we're pulling from dad is still the uppercase G, and then now we can get the lowercase b that is coming from your mom, because that is a possibility. All right. So there we go. Are we done yet? No, we are not. And I know that you are so excited that we are not done with this yet. Because your dad could give you a lowercase g. Okay? So let's just put a lowercase g here. I don't like how it's not recessive or dominant first, but it is what it is. So your dad is giving now that lowercase g. All right, and now we're just going to say, your mom could give you an uppercase b. There we go. 50% from your mom, 50% from your dad. All right. The last one too, are we done? No, because this lowercase g can also be paired with the lowercase b. So G and B. 50% from mom, 50% from dad. Okay. I'm going to leave some time on Monday to kind of re-explain this, but that is how you're going to get, you can have 4 different possibilities, okay? And maybe I want you to kind of just think about that as each cell, this is an oversimplified version, each sperm cell still has half the genetic information from your mom, half from your dad. It gets swapped back and forth, okay? So, moving on to the next part. You do need to understand this, okay? The idea of dihybrid crosses. You're going to have to pay attention to me on this, all right? So, dihybrid crosses, you're going to have to go, it's going to be kind of a little bit slower here. This question is just a little bit different, but that's okay. But we're going to say a male rabbit, which is a genotype of capital G, 2 lowercase b's, is crossed with a female genotype that has 2 lowercase g's, uppercase b, lowercase b. On the test is going to be set up for you, okay? You're not going to need to set this up at all. You're now going to need to fill out each box one at a time and then figure out what genotype is with what genotype. And my pencil better make this to the end. So, how we do this is we're going to go one at a time. All right? So we're going to look right here. Let's just start up top just to make life easier. We're going to start with this one. First letter is the uppercase G. Uppercase G is going here. Okay? Now we're gonna work this way now. Okay? I don't know, that's a weird one. Now we're going to work this way. Okay? So we're now going to take the first letter, which is lowercase g, and that goes there. Okay? So that's how we get our first one. G, G. Okay? Now, I think you see where we're going with this. Let's just take the one up top. That lowercase b is going to come here now. The uppercase b is going to slide here. Okay, so I'm going to go ahead and try to clean this up to the best of my ability without deleting too much. And that is how we get that one. All right, let me kind of clean that up some. So B in there. There we go. All right. You can either work down or across, but hey, let's just let me figure out which one I want to do here. Let's just work this way. So we're going to do this one next. Okay. This one, same way, Keith. Take that first one. That's where that G comes from. Take that first G. That's where that goes. Boom. Just like that. All right. So we have that. Now let's work towards our second one. B is going to go here. B. That's where this one comes from. All the way down. Okay, there we go. The difficult part, or the one part that's probably going to take the most time, is you're now going to have to look at each square, and you're going to have to figure out, all right, what the phenotype would be. But you already know how to do this because we have had a lot of practice. So I see there's an uppercase G. So this means that this box is going to have gray hair, all right? I also see an uppercase B. So that means if we look up here, that means we're going to have black eyes. Okay, so gray and black. So you're going to have to fill out this entire box and then go through each one very carefully. And then all you do is just count out how many are gray and black. I don't know. Let's say 4. And then maybe each one of these has 4. I don't know. You have to figure that out. But that is how you do dihybrid crosses. All right. Now, that being said, you say, all right, now I know how to do that, Mr. Billick. Now you go to the dihybrid cross practice. From here, just like you did before, there's only 9 questions. Do these questions. Try your best. It frustrates me when you don't even try. But the reason that I have this is for you to be prepared come test time. Okay? So you know what's on the test and you're not surprised by anything. So after, like I said, what I want you to do, and I'll even explain to a little bit later, is let's pause this video now because I am going to explain about the gizmo next. Pause the video now. Work on this dihybrid cross. So you should be paused. Okay. All right. Now, this is a terrible assumption. I'm assuming that you all have already finished the dihybrid cross because I told you to pause it and work on it. If you haven't started on the dihybrid cross, pause this video now and work on the practice. Okay? Okay. Now let's hope everybody did it. Wishful thinking. So, after you finish the dihybrid cross, what you will be doing next is you will be working on the mouse genetics with one trait. Alright? So, the mouse genetics with one trait gizmo and the mouse genetics with one trait doc as well. I have a link included for you all because I didn't put it in there before. So let's click on the link first. Do not try to, the gizmo won't pull up in here. Click on this link now. Check that out. It opens up a new tab. All right. Let's go to Blue 1, for example, and gosh darn it, what I meant to do is let me go back. My classes, whatever. Let me add one real fast. Genetics, I already know which one I want. Bio, genetics, mouse with one trait. 1, 2. Did it do this one? Sure. Okay. All right. So you now, it should look something like this. Okay. So I want to explain the gizmo first a little bit. So we're going to click on the mouse genetics with one trait. You need to click launch the gizmo. It will look something like this. Okay. So there's a couple things that you need to know. You can drag and drop these mice. You can drag and drop 2 of them. All right. There's the clear button. You can drag and drop both of these. There's a breed button that you can click multiple times. If you need to reset anything, you can then clear it. All right. So let's say we brought these 2 mice together. Now let's say, hey, let's look what else we have. If we click show genotype, you just need to maybe not click on the mouse, or just hold it over the mouse, and it will tell you right underneath here. Lowercase f, 2 capitals F. You can also do a statistics. It can also show you in percent. You are going to need these, okay? These tabs open to finish up the doc. All right. So let's say after you breed a whole bunch of them, there are also what you need to know down here are these holding cages. So let's say we want to take one of these mouses and another one of these mouses as well. Okay. Then we can do is, hey, let's clear these, and now I want to breed these. You can just pull these up, and you can now see what the genotype is. So if we click breed, look at that. Now we're getting different colors. All right. The gizmo is super simple. All right. It really is. The gizmo is ridiculously simple. So that's a simple gizmo. You just need to be high school students and read the instructions. Okay? You all have done this a couple times now. Read the instructions. All right? It says to click clear. It says drag and drop certain mouses. Don't click breed yet. Read the instructions. All right? And that's what you're doing today. That's it. I'll go back to here so we can kind of see what the plan is. First things first, do the dihybrid practice. You can take it 3 times. It does not count for anything. This is for you to be comfortable. Then do the mouse genetics with 1 trait. And that's what you're doing today. Don't be turds for the sub, and I will see you on Monday. Adios.